Hello again guys, and welcome to the channel. So for those of you that are following my progress, you know that I am mainly putting out videos regarding molecular biology and basically how-to videos in genetic engineering. So I wanted to make this impromptu video for you, just to do a recap on what happened in 2022 and maybe share the plans for 2023. So 2022 has been a good year for me actually, since I did manage to test all of my plasmids for optimal expression in mammalian cells. The results that I got, which are explained in more detail in this video right here, are going to be published actually in a real scientific paper. So my article, regarding Golden Gate assembly and the whole method for building plasmids will be published. And that is a huge deal for me. And going forward in 2023, what is actually noteworthy is that I did change the lab. So now I'm working in a lab in a different floor here at the university. If you have time, like 1 minute, I'll do a quick tour on it. So I do have everything that I need to keep working. And what exactly am I working on in 2023? Well, the plan is as follows. I am trying right now to develop an antibiotic-free selection system for plasmids. So what this means is usually when you have plasmids and you want to grow them in bacteria, you usually need to use antibiotics. But right now I'm trying to avoid that, and hopefully this will increase expression in mammalian cells also, because in 2023 the second thing I want to do is basically try out the GBA gene in mammalian cells. So for those of you that don't know, the GBA gene is lacking in a disease that's called Gaucher's, and my main focus right now is to develop a gene therapy for that particular disease. So yeah, we'll see how it goes, and I'll keep you guys updated as I usually do. So as promised, let me show you what I got here. I'm gonna tell you what I am using, and stuff that are not mine actually, because of course it's a shared lab. So this is my sink right here. This is a hood, really nice. Yeah, it's smaller than the other one, but it suits me just fine. These are some real-time PCR machines and whatnot. What can I do? They're just here. Plates just here, not mine. But however, what is mine, I've managed to purchase one of this. I got it in installments, but yeah, this is a mini PCR. Okay, this is my my tray right here. So here I have some sterile tips. Here I got some tips, and here I got some miscellaneous stuff, like gloves, reagents, and so on. Some tubes. Got some distilled water, LB, and stuff like that right here. So this is the centrifuge that I'm using. Yeah, it's loud as hell. And a little scale. This is also mine right here. So this is my electrophoresis tank. And of course tips, and I have a transilluminator right here, and this is the power source for the electrophoresis. Yeah, so these are mine. So yeah, pretty happy with the setup right here. Hopefully it's gonna suit me extremely well.